Good afternoon, students. Today I am going to continue about the process of osmosis. Already we have seen the experiment on osmosis, that how this process takes place. Like one experiment also I have shown to you. The water molecules move from lower concentration to higher concentration when both are separated by cellophane paper or egg membrane or animal bladder, etc., which acts like semi-permeable membrane. So today I am just going to tell the the rubber sheet and muslin cloth instead of cellophane paper. So in the same experiment I can show you here that here it is shown the cellophane paper. In this place rubber sheet is tied here, and that act as a barrier. Rubber sheet is not having any tiny pores which will not allow the molecules from lower concentration to move to the higher concentration or from higher concentration to lower concentration. So there will be no net movement of the uh, water molecules from lower to higher, and no rise in sugar level will take place. And the next is muslin cloth. Instead of this cellophane paper, here it is tied the muslin cloth, which acts like a barrier, we can say, because it is having the larger pore. It cannot hold any molecules. So the sugar molecules from the thistle funnel will come down due to gravity and mixed with the water. So there will be no net movement, no rise of sugar level will be shown. Means no osmosis takes place here. And the next same experiment can be performed by using a whisking bag. Here you can see a whisking bag, and whisking bag is acting like semi-permeable membrane. So in a beaker the water is taken here. In whisking bag sugar solution is kept there here, and in the whisking mouth of whisking bag, capillary glass tube is inserted and tied it tightly. Now this setup is left for few hours, and the initial sugar level is marked there and kept for few hours. After few hours we can see that the rise in sugar level, that means water molecules moved to the whisking bag, which contains the sugar concentration, which is highly concentrated than the water. So the when it enter, water molecules enter here, which help in rise in sugar level, and the water level comes down in the beaker. So we can say here the osmosis is going on. Next, two key points in the above experiment whatever we have seen. First is regions of different concentration of water molecules, or two regions of different concentration of sugar molecules. Second, the two liquids are separated by a semi-permeable membrane. Here, two regions are separated by a whisking bag which is acting like semi-permeable membrane. A semi-permeable membrane is one which allows only certain molecules, smaller molecules, to pass through it, like water molecule is passing through the whisking bag to the whisking bag, and the larger molecules will be held back. So here the osmosis continues. Next, we see here the semi-permeable membrane and two types of solvent, that is, here more dilute solution is kept here, and here less dilute solution. So the movement of water molecules from less dilute solution to less dilute solution to from more dilute solution to less dilute solution, means from lower concentration to higher concentration. You can see the moving water molecules from lower concentration to higher concentration, but sugar molecule doesn't move to the lower concentration side. So osmosis is taking place here. How long can osmosis continue? Now, how long the osmosis can continue? For this, we can say that there are two types of concentration here, lower and higher. So, till it become uniform, the osmosis will continue. But here we see when sugar level rises up, it reaches the height. When it reaches the height, it is having the weight and pressure. When weight and pressure is there, it stop the osmosis process because it put the pressure from there and the osmosis cannot continue due to weight. Pressure is exerted here, so the osmosis cannot continue even though the solutions in the both in the beaker and whisking bag is not uniform. So osmosis cannot continue so long. There is a limit. When it reaches the height, it put the pressure, and due to pressure it will not allow to continue the osmosis. Osmotic pressure. Here the same experiment, in that little changes is there, that weight is put, yeah, a piston is inserted in this thistle funnel. Airtight piston is inserted in this thistle funnel with weight. So, though the concentration in thistle funnel is highly concentrated and beaker is containing the water, but the movement of water molecules to the thistle funnel will not take place due to weight is put with airtight piston. That's why pressure is there, weight is there, and it doesn't allow water molecules to move through this cellophane paper to the thistle funnel. No rise in sugar level. It remains as initial level. So there will be no osmosis taking place here. Dear students, here how to define the osmotic pressure. Sometimes you will be asked to define osmotic pressure. Osmotic pressure is the minimum pressure that must be exerted to prevent the passage of pure solvent into the solution when both are separated by a cellophane paper or semi-permeable membrane. So when weight is put, that is preventing the pure solvent to pass into the solute. That is osmotic pressure. Please learn the definition and go through the chapter and please solve related to this question answer. Thank you.